Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Willy Maulana Hibban. I'm going to be a moderator in this presentation. We are group 6 and we are going to presentate about environmental influence on plant disease. The members of group 6 are Elsa Nuraini, Pramesthi Mahanani, Khalifatu Rabbi Hadi, Willy Maulana Hibban, Aisyah Rizta, and Adika Trias Pruwita. In addition to susceptible host plant and virulent pathogens, environmental factors are also factors that cause disease in plants. These environmental factors can help or inhibit the development and spread of pathogens.
For example is number 1, blackroot disease on tea in young still. Number 2 is plague disease in maize only in the lowlands. Number 3 is smallpox in tea in the high plains. Next. Environmental factor can influence through pathogen and host plants. Next. By influencing the pathogen prior to infection as long as the pathogen is still present outside the plants. After the occurrence of disease, after the pathogen is in the plant's body, the multiplication of many pathogens determined by external or environmental factors. Next.
By influencing the host plant, plant resistance and susceptibility to disease is not a permanent thing. Example is applying too much fertilizer can make plant more susceptible to disease. External factors can influence individually, but also can interact with each other, affect both pathogen and host plant. Example is optimum temperature for pathogen growth is not always cause the growth of severe disease because this temperature can also have development plants so that plants have a resistant higher.
Next, weather influence. The influence of weather on plant disease includes humidity, weather, sunlight, and acid and carbonic acid. Next. First, humidity. Influence on spore development. Moisture affect the development of spores. Some spores are able to germinate at high relative humidity because spores mostly have low water content. In dry air humidity, conidium will die within 1 until 2 hours.
High relativity inhibits transpiration, thereby reducing rate of transport of nutrient solution from soil to soil plant organs. Potential condition for the emergence of pests and disease or for the development of pests and disease. Increase the development of spores and pathogen infection in plant. Difficult for post-harvest handling, namely the number of diseases that attack crop yields, especially in storage areas. With low cases, the transpiration rate of sometimes excessive so that it can cause the leaves to will temporarily until the flow of water from the roots can compensate, causes the development of insect factors to be higher, resulting in disease caused by virus. Next. 2. Weather. Temperature can affect the disease, namely slow down or speed up. Temperature can affect the number of spores that germinate. Spore germination speed and germination type. Spore germination mean 1 until 3 degrees Celsius, max 30 until 36 degrees Celsius. Opt for the type of mushroom. Low soil temperature will result in distribute absorption of water and nutrition because transpiration increase. If there is a continuous lack of water, the plants will be damaged. The relationship between low soil temperature and dehydration in plant tissues is when the plant temperature is low. The viscosity of water increase in the cold membrane, so that the activity of physiology of root shall decrease. Next.
Number 3, sunlight. Indirectly after humidity directly affect the pathogen which is still outside the plant body. Like with an intensity of plus minus 400 lux, inhibit conidia germination by 30% compared to that incubated in the dark. Plants may show other symptoms when they are not receiving the required amount of sunlight, such as yellowing of the plant leaves, pale coloration, small leaves, and poor growth in addition to failed flowering and long, thin stems. Without sufficient light, plant will experience decreased growth and die due to their inability to produce food through the photosynthesis. Number 4, acid and carbonic acid. The effect of less significant because in the air the leaves of substance is has changed mass. In general, fungal spores cannot germinate in anaerobic state. Spore germination and respiration of Aspergillus niger instead it is accelerated by carbonic acid to levels of certain. It is suspect that in the metabolism of this substance it becomes precursors of amino acids.
Yeah, this is the last slide, and I'm gonna say thank you for watching and learning from our presentation. If you can, text it on discussion forum at Spada, and have a nice day. See you.